Hello, this is Mrs. Kasperwitz, and today we're going to be taking a look at designing a controlled experiment. This section of your notes should be pages 7 and 8 in the Unit 1 note packet. On each slide, you're going to see red highlighted words or terms that you are going to be recording on those pages, kind of in a fill-in-the-blank manner. So let's get started. So in a controlled experiment, you have to have at least two groups of test subjects. One group, the experimental group, is going to be the group of test subjects that is changed in some way. So in other words, it's going to be receiving the factor that the researcher is testing, which we refer to as the independent variable. So an experiment needs to have at least one experimental group, but there might be multiple experimental groups. So for example, you might want to change the amount of fertilizer a plant is getting, and you're going to have 2 or 3 experimental groups. Each experimental group is getting different amounts of that fertilizer. In the ants can count experiment, we had 2 experimental groups. The one group of ants that got the stilts added, and then the one group of ants that had their legs cut off. So again, this group, the experimental group, will test the independent variable, which is the factor that's changing in this group. So one of the ways that I tell students to help them remember the independent variable is that it's the factor that's change in, okay? Because it's the independent variable. So maybe that will help you remember the difference between an independent and a dependent variable in an experiment. And like I said, there might be more than one experimental group in an investigation. So in this little investigation of plants, we have 3 different experimental groups. We have each of these groups receiving a different color of light. Now, this is extremely important, which is why it's got a little tag next to it in your notes that says important. There should be only one independent variable in an experiment. So in this experiment here, where we're adding again some fertilizer to some plants, there's only one thing that we're changing in each of the experimental groups, which is the amount of fertilizer. So for example, in this picture, we see there's 2 drops of fertilizer. This one has 4, and this one has 6. So the only thing we're changing is the amount of fertilizer being added to each of these experimental groups. In the ants that count experiment, the factor that we were changing was the length of the legs of the ants. And so there was only one independent variable. We had 2 experimental groups, and the thing that we were changing was the length of the legs. So a controlled experiment must also include a control group. This is the group that's kept under normal conditions. So this is the group that we are not going to change or give the independent variable to. In a controlled experiment, there must be a control. So there has to be a group that we can use to compare the results to. So this is what happens in the normal situation. This is what happens if I change a factor, and then I have to compare those 2 things. So I can see if the factor that I was changing, the independent variable, is actually having an effect. So the results of the experimental group are then compared to that control group. Right. And so we look to see, is there a significant difference between what I saw normally and what I'm seeing when I change the factor that I'm changing? And if there's a big difference, then we can say that that factor had an effect. And if there's not very much of a difference or none at all, then we know that that factor is not something that is important in the outcome. So in our little plant experiment here with the fertilizer, this is the group here with no drops of fertilizer extra that would be the control group, all right? Because this is the one that's under normal conditions without the fertilizer. So I can go and compare my experimental groups back to this one and see, is there a change in the height of the plant? In the ants can count video, the control group was the ants that were regulars. All right, their legs were not adjusted in any way. Experiments must have a control group. If they don't have a control group, then you don't have a valid conclusion. So, for example, in this little plant growth investigation, we don't have a control group. We have different colors of light, but we would need a normal light situation, which is white light, or dark, to compare these plants back to as control groups. There can be more than one control group in an experiment, but for our purposes, we're going to just stick to just one. So, in addition, controlled experiments should have as many constants as possible. These are the factors that remain the same in both the experimental and control groups. So we really only want to change one thing between my experimental and my control group, just one, and that's the independent variable. Everything else, as much as possible, we want to keep the same so that there's not some other possible factor affecting our outcome. Right. So the more constants we have, the more valid our conclusion is going to be. So the more things that we can keep the same, the more likely it is that it's our independent variable. It's the thing that we're changing that's affecting the outcome. So if we take a look at our plant experiment here, what are the constants? So take a moment and think about that for a minute. So if we look here, we can see that all of the plants are being given the same amount of water. All of the plants are given the same kind of light. All of the plants are the same variety or same species. They've all been planted in the same type of pot, and presumably the same type of soil is in those pots. So those things are all being held constant, so that the only thing that we're changing is how much fertilizer is being added. So we want to isolate that factor from all of the other things that need to be staying the same. So once we've got the experiment set up, then we're going to run it and we're going to collect some data. And that data is the dependent variable. So the dependent variable is what you're going to measure during the experiment. And this is what will become your data. So the dependent variable is dependent on the change that we've made. Okay, so we change the independent variable, we change that factor, and then we look to see what the outcome is. And it's the dependent variable that's the outcome, right? And we're going to measure that outcome either with numbers, quantitative, or observations, qualitative. So measurements would be, taking the temperature of something, measuring its length, measuring how big it is, something like that. And the qualitative data would be like observing a color change, observing a smell, does it bubble or not? Those are qualitative things. And in a good experiment, you're going to probably want both of those to be there. So what could the dependent variable be in this experiment? Let me get myself out of the way here for a second. Okay. So in this experiment, we might want to have the height of the plant be the outcome that we're measuring. Maybe it's the size of the fruit that is being produced by that plant. Maybe it's the number of leaves that grow on that plant, but that's something that we could measure, the dependent variable. Okay. Now, that's the basic stuff that we need to do for a controlled experiment. We need an experimental group, we need a control group, we need to have one independent variable, we need to know what we're measuring, the dependent variable. We need to have a whole bunch of factors that are staying the same in all of my test subjects in both my experimental and control group. And then I'm going to take a look to see what my results were. But there are some other important things to think about while we're doing some experimental work. So again, let me scoot myself over here if I can. and we'll take a look at sample size. Whoopsie. Okay, so our sample size is really, really important. And our sample size is just the number of test subjects that we have in each experimental and control group. So we want to have a large enough sample size that it represents the larger population as best as possible. So the larger our sample, the more representative it will be of our larger population. So in essence, then bigger here is always better. The more test subjects we can have, the better our result is going to be. So for example, if I can't study all humans, right? So right now we're in the middle of this pandemic and we're trying to study this new disease, COVID-19 virus, and we're taking samples of human beings that have had the virus or been infected by the virus, or when we're getting ready to do the vaccine trials, we're picking a small group of humans to be our test subjects and test this new vaccine, right? But we want that sample size to be large enough that it will actually reflect the entire population. We don't want to just pick 10 people to test the vaccine on. 10 people is not large enough to represent the 7+ billion people that are on this planet. So we're going to need thousands of people to participate in any sort of vaccination study because we want that population to be represented by the sample. Okay? So sample size is really important in an experiment. Okay? So the bigger the sample size, the more test subjects we have, the more valid our conclusions are going to be. All right, so we want to pick a large enough sample size that we get a good, valid conclusion. If we can't get a large sample size, then sometimes what we'll do is we'll have more than one trial. So we'll repeat the experiment more than once, all right, to increase the amount of data that we've collected. So again, that's a way to gather data from a small sample size, is do more trials. So you're going to repeat the experiment more than once. Some other things to consider, all right, you got to make sure you know what you're doing. So you got to consider what materials are going to require. Are there any special safety precautions that are necessary? All right, we talked about those in lab. You have to think about your procedure, right? So you need to think about what you're going to do. So somebody at the end of this, remember the peer review part, somebody has to be able to like redo your experiment and get the same result. So it's important that you, when you're running your experiment, that you're able to have somebody else repeat it and do it again. Right? And you got to be specific when you're doing that because your procedure is like the recipe for your experiment. All right. Other people want to be able to repeat it exactly like you did. So if you are not specific enough and you don't say how much, say baking soda you put in your chocolate chip cookies or how many chocolate chips you put in, then you're affecting the overall outcome. So you got to be specific. You have to also think about how you're going to analyze your data. So when you're done, how are you going to visualize your results? So these are some must knows, all right? Very, very important. That's why they're highlighted must knows, all right? So a controlled experiment can always be improved by adding more test subjects or by increasing the number of constants. So adding more test subjects will give you a better representation of the population as a whole. And increasing the number of constants is going to mean that you're changing only one independent variable so that when the outcome happens, we know it's from that one thing that you wanted to study. It's not from something else that may have affected the outcome. So increasing the number of test subjects and increasing the number of constants will always make an experiment better. So you can look at this in the opposite way and say, what are some possible sources of error in an experiment? Well, one is not having a control group. So if you're reading through an experiment and we don't have a group of test subjects that is under normal conditions, then we don't have an experiment, right? It's not that you can't get some useful information from what you're doing, but it is not considered to be a controlled experiment without that group. Again, too small of a sample size. So if you're reading a question and there's less than 10 in the sample, that's a pretty good chance that there's a source of error there, and that's a way too small sample size. And lastly, more than one independent variable. So this is sort of the opposite of having more constants. So if you just want to have one independent variable, you have to up the number of constants that you have. So that's a possible source of error if you have more than one independent variable. So say, for example, you're studying the effect of light intensity on bean plants. So you want to change how bright the light is and see how it affects their growth. But you also have one of the groups is getting more water. So now you've got a problem because the bean plants that are in that group could be growing taller